This is sound localization and sensory habituation in Teleogrillis oceanicus. And before we get started here, I just want to explain a little bit about sound localization and sensory habituation. These two concepts are pretty crucial for you to understand going into the more complex information in this study. So, sensory habituation, the best way that I can explain it in simple terms is that there is a constant stimulus over time, and the neural receptors are going to be having a decreased response to that stimulus. And that might be a little confusing at first, but I'll give you an example. I moved into a new house a few months ago, and it's on a busy road. And whenever I first moved in, the road noise was driving me crazy. I could hear it all the time. It was constant, and it was always eliciting a response from me. Something sort of like this, what this girl's doing right here, covering her ears and screaming. So the noise is getting a response from me. I'm getting annoyed, I'm getting bothered, I might even decide to go somewhere else to study, and so forth. Now, after just a little bit of time, I started to get used to it, and it didn't give me the same response. In the term that we like to use is we tune it out. Just tune them out. Somebody's complaining to you, just tune them out. So the road noise has started to essentially tune it out. It's not the same response. I'm not getting the same response. I don't even notice it anymore unless I actually think about it. So this is kind of an example of sensory habituation. There's a stimulus, and at first I am having a response to it, and as time goes on, I am no longer having the same response, or this response might be diminished. And this next slide is showing the difference of, this one is showing us sound localization. And this is using a human's head. So we have our two ears, left and right, and we have sound coming from one direction. Of course, the sound is going to be hitting the right ear first, and then the left ear on the other side second. And so we can see that with this little chart here, hitting our right side first, and then the left side a little bit later. And that just makes sense if you think about it a little bit. And then over here on the amplitude, it's going to be louder on the right side than it is for the left side. And this gray part is a sound shadow. And for what we're talking about today, which is crickets, it isn't really important. But if you want to learn a little bit more about that, you can look it up. But for what we're talking about, not too important. So like I said, we are talking about crickets. The concepts are still the same here. The mechanisms are just a little bit different, but these concepts you need to understand before we go into the rest of this. Okay, so we are talking about Teleogrillis oceanicus, and these are a type of cricket. And something about crickets that you may or may have not known is that their auditory receptors are on their hind legs. So that is just a little bit of information for you. It doesn't really, matter for the rest of this study. But their auditory receptors do experience habituation, and habituation occurs over a chain of stimulus for a certain amount of time. And so we're going to be looking at the same side ear where the stimulus is occurring. So it's called the ipsilateral ear. Ipsilateral means the same side. So when the sound source is coming from one direction, there is a greater intensity at the ipsilateral ear. It is louder at that ear, of course. So therefore, the habituation response will be greater for those ipsilateral receptors. It's just a little bit of a broad overview for you. We'll go into more of what this means and some proof about this and what this does for our sound localization in the crickets. So for methods, I made a little diagram here. You just have to use a little bit of imagination. This is a cricket on like a tray that they have, held the cricket down We strap the cricket down on this tray. So you just need to use a little bit of imagination. We have the body here and then the two hind legs here. Like I said, these receptors, these auditory receptors are on the hind legs. So what they did is they cut into the legs, placed some electrodes, and that is how they are going to be measuring the neural responses. Now, the other part of what they did is they created the song trains that closely resembled crickets in the same species. So they would play the songs to the crickets on that ipsilateral side and measure the response. And they would do this at different levels of sound and at different pulses. And so these crickets, they send out their songs in pulses. So we'll see a difference in pulse rate and a difference in intensity. So this is figure 1, and this is showing a decrement of responses to rapidly repeating stimuli. This is showing us our habituation. And 1A is showing us a single auditory receptor, while 1B is showing us the whole nerve receptor. The single auditory receptor is pretty hard to use because you can't use it for a full hour. So they're playing the songs for 30 to 40 seconds with 60-second rest in between for a whole hour and measuring the response. The single auditory receptor will not last that long. It'll only last a couple of minutes. So what they did is they used single auditory receptor here up top and then compared that to a whole nerve down here and said, hey, these are really similar. So for the rest of the study, we are going to be using whole nerve recordings, and they're so similar and they represent the same stuff that it's okay. So, but we can go ahead and talk about what this is talking about first. So we have our spike count on our y-axis and our time on the x-axis. We're looking up at A, and so what they did is they start the sound, and the very first thing is a high spike count. We're having a high response, which is represented here by these spikes, and also here by the point on the graph. Now, as time goes on, we see quite a bit of a decrease, and by the 10-second mark, our spike count is much lower, and we are down in this range, kind of flattening out with our response, which is not as much and not as much as it started. The exact same story is true for the whole nerve down here. And so we have at the very beginning, t equals 0, time is 0. When we initially started the sound, there is a great big response. Spike count is pretty high, and then as time goes on, spike count decreases, the relative response is decreasing, and we're ending up down here. In the paper, they say that the asymptote for these two different pieces of information are so similar that it is a-okay that they're going to use the whole nerve cell for the rest of the study. But this also shows us a great example of habituation that we see in the cricket. So figure 2 is a little bit more complex, but we're going to talk about it piece by piece, so it's not so daunting. Figure 2 represents the decline in response strength for different pulse rates and intensities. So this is very similar to figure 1. We just have a little bit more going on with, we can now see the pulse rate, which is up here in this corner, and it changes for each one. And then we can also see the intensity in decibels over here. So let's talk about this first one right up here. So our pulse rate is 1, okay? We have a pretty low pulse rate, and then we have our intensities here, and these are in different colors. So the 100 is corresponding with green, 90 purple, 80 red, and so forth. And so we have low pulse rates, and even with a high intensity, we are not getting any decrement. Everything is pretty flat. We are getting the same amount of response throughout time. And so that is what we're measuring over here on our Y as a response, and then our X is time. So response, it's the same throughout. We are not getting any habituation. As we move forward, we're starting to see a slight decrement with the 100 decibels, 90, and also 80 whenever we are using a pulse rate of 8. So we start to see slight decrement, and this is slight habituation. It just gets even quicker as we go to 16. Our pulse rate is increasing. Now our decrement is much steeper, and we start to see a little bit of a decline with the 70 decibels as well. And as we go to the last one with it, which is 28 for the pulse rate, it is the quickest to decline, and everything is rapidly declining except for this one down here, which is 60 decibels. And so what this is showing us is that at a high pulse rate and a high intensity, the decrement will be much quicker. You'll have much quicker habituation when you have a high pulse rate and a high intensity. And so this habituation causes an increase in response latency. So our response is going to be later. The interaural latency difference alone can be dangerous, but can also provide directional information. The problems that occur with high intensities and high pulse rates are that the response can habituate so much that you don't even get a response at all. And so there can be a danger with that if there's some sort of threat nearby. And so, but you know, actually in nature, if you were seeing this, the cricket would be close enough to the danger to actually just see it. So it really isn't that big of a danger. So what's pretty interesting about this is the localization cues from the interaural latency difference. And these appear to be far more reliable compared to response strength. As we showed in the past slides, the response strength is decreasing with the stimulus, that it is habituating. So the localization cues that are coming from the latency differences, those don't change. So those appear to be the most reliable as far as learning which way the sound is coming from. So this was tested in a lab where the cricket is held to the board. Well, in nature, crickets actually do this kind of zigzag walking path, and they're thinking, hey, this might actually be because of this habituation, and it might help to minimize the distortion caused by habituation. So in conclusion, the increased pulse rate and increased sound intensity lead to habituation in T. oceanicus. Sensory habituation leads to response latency. Response latency is a reliable way to determine sound localization. And linking this into why it matters, they're thinking, hey, maybe this is actually occurring in vertebrates that do similar song-like calls, like birds and frogs. So they might actually use this for sound localization. And that is the end of this presentation. Thank you.